Enzyme, a substance, usually a protein, that speed up a biochemical reaction by modifying specific substance called substrate. Imagine a scenario. Consider a person. She don't even have a sponge to clean up the dish, and all this time she just did it by hand. She did clean the dish, but she need a little bit more time. And one day she got a sponge, and after that her dishwashing chores become more and more efficient and faster. That scenario is basically how enzymes work in our body. The dishwashing activity is the same as the reaction. The unwashed dish as a substrate, the washed dish as a product, and the sponge as an enzyme. And the fact that the person can wash the dishes faster once having the sponge, so does the chemical reaction after receiving the enzyme. In a way, we could consider enzyme as a tool for reaction to go faster. But in enzyme kinetics, there are explainable reasons as for why does, in the presence of enzyme, the reaction become more efficient. This is because the activation energy of a reaction is lowered in the presence of enzyme. Activation energy is the amount of energy for the reaction to begin. Think of a car that wants to drive uphill. It requires a lot of fuel to climb the high hill. But if the hill is just a low one, it will require lesser amount of fuel. Thus, the reaction goes faster. An enzyme can create the product either by breaking the substrate or build up the substrate. Enzyme will not be used up in the reaction and can be used over and over again, like a sponge in the early analogy. In enzyme, there is a place called an active site, where the substrate will bind and the reaction will occur. One key characteristic of an enzyme is the active site is highly selective and specific, which means a certain enzyme will only work on a certain substrate. Active sites are also very sensitive. The slightest change in the environment will affect the enzyme activity. There are several factors that will affect the enzyme activity. The first one is the temperature. Enzyme works the best around 37 degrees Celsius at body temperature. Extremely high temperature will denature the enzyme. The second factor is pH. Enzyme work the best around pH 7. If the environment goes too acidic or too basic, the enzyme will be denatured. And the third factor that will affect the enzyme activity is the concentration of the substrate or the enzyme. In this home-based experiment, we will run an experiment to observe the effect of environment on enzyme activity, particularly on temperature and the substrate concentration.
So we are going to analyze and discuss the experiment that we have done before. So this experiment is basically we are testing the enzyme activity on the protein. So we are using the meat tenderizer that contain bromelain as enzyme and the gelatin that contain the protein as the substrate. Meat tenderizer contain bromelain, which is a mixture of enzyme that digest protein. This enzyme is classified as proteolytic enzyme. So gelatin is made out of an animal protein known as collagen, which is structured in triple helix. For the preparation of the gelatin, when we add hot water to the gelatin, its heat causes the long chains of protein to unwind. As the gelatin cools and solidifies the collagen, it reforms messy and tangle, which trap water in the middle of the long chain proteins, turning what started as a liquid into a wiggly mess of semi-solid protein. So when bromelain makes contact with gelatin, it begins to break down the peptide bonds in the collagen protein as the long chain protein are cut up into smaller protein. They gradually release the trapped water, returning the solution to a liquid state. So for the experiment, we divide it into three parts. The first part is the basic parts, which is we just add the meat tenderizer into the gelatin, and then let's the reaction okay. The second and the third part is we are studying the factors that affect the enzyme activity on protein, which is the temperature and concentration. So the result that we gain from the experiment for the first part is the cloudy region become bigger. The result for temperature is the thickness of the solution occurs, but after several minutes it stops. The reaction stops. Okay. And for the third part, the concentration of substrate, it takes a few hours in order to decrease the thickness of the solution, and then it stops. So let's discuss. For the first part of experiment, that is the basic overview. The reaction of the bromelain and the gelatin does not occur immediately, but it occurs slowly. If we left the bromelain in contact with the gelatin, the more proteins will be broken down and the more liquid will be left on the surface. So we want to make it faster, we shake it and stir it, and then we can see that almost all of the semi-liquid change to liquid. For the second part of the experiment, we are studying the effect of the temperature on the enzyme activity. So the rising temperature generally speed up a reaction, but extreme high temperature can cause an enzyme to lose its shape and stop working. So at boiling temperature, bromelain breaks down and will no longer be able to digest the protein in the gelatin. So as we observe that the reaction between the bromelain and the gelatin occurred immediately, but it stopped 10 minutes. After 10 minutes, it is because the enzyme, the bromelain enzyme, has been denatured, and therefore it has stopped working after 10 minutes. So enzyme work best within the specific temperature. The effective temperature range for the bromelain is between 40 to 65 degrees Celsius, and with the optimum is being at 50 degrees Celsius. So the boiling point temperature is about 100 degrees Celsius, So here we can conclude that the enzyme is being denatured, and therefore the reaction occurred immediately, but it stopped 10 minutes later because the bromelain cannot working again. And then the last part of the experiment is we study the effect of concentration of substrate on the enzyme activity. So once all of the enzyme have bound, any substrate increase will have no effect on the rate of reaction. Due to the limited number of enzyme molecules that can be interact with substrate by the case, there is a maximum rate upon which it works, and so there is a certain limit where if we keep increase the concentration of the substrate, the rate will not keep increasing as well up to a certain point. So as the available enzyme will be saturated and working at their maximum rate. So as we can see that it takes a few hours in order to decrease the thickness of the solution, and then it stops. It is because the number of enzyme molecules is not enough to interact with the substrate.
This experiment was conducted to observe the effects of environment towards the enzyme activity, and we chose temperature and substrate concentration as our main condition out of few other factors. There are no fancy equipment since this is just a home experiment, and we also select the material from the things that could be found inside the home. The enzyme we chose is bromelain and the substrate is gelatin. For the temperature as a factor, we could observe that as the temperature increases, the reaction rate will also increase, but extremely high temperature can destroy the enzyme activity. For substrate concentration, if the substrate concentration is high, the reaction rate will also be high, but only until certain concentration point. Beyond that, the rate will be constant as the number of enzyme is limited. So, to conclude this home experiment, we could proudly say that this experiment is a success and the results obtained are as expected.